the ideas behind the concept of targeted metabolomics, which was instrumental for more than 12 years for the foundation of the Biocrates company. And I have made a title because I think the technology and the concept we are introducing today has a major strength in the study or to study the interactions between genes, environment, disease, and physiology. So I have made a title, Metabolomics as a key tool to explore the genetic environment health paradigm. The challenge is that we are dealing with, in an overall side, we are dealing with rather poor or low drug response rates across a wide range of indications. Still, this is most pronounced in cancer, in oncology, but it's also true for several other indications where drugs are used. And of course, personalized medicine means to get these statistics to a better range. And the tools to achieve such goal are, of course, as you all of you know, to get biomarkers, to get better phenotyping of patients, to identify subclasses of patients, to get better stratification for patients, and nonetheless, from a mechanistic point of view, to get target-oriented therapies, on a mechanism-oriented therapies. It illustrates the wide range of areas where biomarkers have use. So most common are clinical disease markers, but we have already a lot. We have a few mechanistic markers, in particular in oncology, which are utilized in companion diagnostics. And what we need much more are therapeutic markers, which enable us to carefully monitor therapies. Why got we interested in so-called metabolomics? When we started with that idea, the initial project was by the German Human Genome Project already in 1997, where I proposed a project to screen ENU mice by metabolomic profiling by tandem mass spectrometry. At that time, the term metabolomics was not yet coined. It was not yet on the market, because we brilliantly could screen by this technology newborn mice. Why shouldn't we screen our patients, our newborns, to improve newborn screening? This operation was able to detect 30 monogenetic disease at birth, even in an asymptomatic stage, out of a single run and a single blood spot of 3 microliters of dried blood. What also have we learned from newborn screening? We have learned it detects by metabolite pattern or by metabolite shifts. We can detect, and that is important, asymptomatic disease prior to clinical manifestation. That's the principle in newborn screening, but it's also very important for metabolomic studies, because it shows that the biochemical phenotype is already evident before any clinical symptoms may appear and prior to the to clinical manifestation. Of course, if you look at genes or look at transcripts and proteomes, they are not endpoints. So information recovered from these sources simply tells what might happen in the future, or is creating a hypothesis. And that distinguishes the metabolome and the metabolites completely, let's say, from the other information sources, in that by definition, metabolites are functionally relevant endpoints. Metabolomic analysis means to delineate functional activity markers, which is unique for the omics platforms. If we know that metabolomics is uncovering really good functional information, one of the main, let's say, application areas is what is generally called in the pharmaceutical development process of mode of action screening. This is the definition. This characterizes the interaction of a compound with its target simply to understand how the compound interacts. So this is a mechanistic with the aim to develop in a very early stage of development a mechanistic marker to guide the further development. This can be done in cell-based assays. This can be done in animal experiments. And the following study, which is several years ago at the starting area of the Biocrates company. So pharmaceutical company wanted to test the technology on this question mode of action, and set out with this diabetes, this type 2 diabetes model, and there was a control group, then an investigational drug or a lead drug of the company, and the active control substance as positive control. And 6 groups of mice, each 10 mice, and 4 samples in the different compartments. Just to note, metabolites can be measured across different tissues or biofluids, because they're more or less in similar. They are more or less the same in these compartments. And at that time, Biocrates analyzed 4 major metabolite classes. And the aim was to this disease model and the intervention to map this to the metabolic space. And I'm only showing a few results. For instance, this is the amino acids in plasma. Here's another result of this study, and showing what the value is of surrogate, metabolic surrogate markers. For instance, the observation was that the active drug is leading to a pronounced reduction of long-chain fatty acids. And in this case, the long-chain acylcarnitines cannot enter the mitochondria, which is seen there, and at the same time, free carnitine is increased because this carnitine is not sufficiently consumed. Finally, you also could pick the differences between the mouse model and the control in by gain ratios. You can rank the metabolites, and you see in this graph these are related to different substance classes. Of course, this is an example, this is a rough proof of principle example of a so-called mode of action study. And the advantages of metabolomics in this sense is that as compared to other, let's say, information levels, one can really map drugs or diseases directly to the metabolic space. That means to the functional metabolic space. And the information which is sufficient to retrieve this content is rather limited and very cheap as compared to other technologies. And that is because we are pinpointing pathways. Pathways are are composed of, let's say, 40 to 100 metabolites, and we only need a few of those metabolites within a pathway if we select them on, let's say, biochemical principles to get the functional information out of that. So that's why targeted metabolomics is a biologically driven concept and approach, and does not request to measure all metabolites over the place to get relevant information. The major application area for metabolomic profiling, in translational search and from a clinical point of view, the early identification of subclinical disease is a central area where we have an unmet need. This can hardly be achieved by existing techniques. We have seen that it's feasible by metabolomic study from newborn screening, where we clearly detect asymptomatic disease by the underlying biochemical phenotype. No symptoms yet at the patient. If we have for instance, a biomarker for pre-diabetes, that would be a big advantage for preventive purpose if we are able to detect subclinical disease. This is that area where, and in that area, if we are digging there, we can delineate predisposition marker for disease or prognostic markers. Okay. This is one thing. The other take-home messages I've briefly touched is subclinical disease risk monitoring. In the pharmaceutical development, it's a brilliant platform to design novel biomarker signature, which give mechanistic information. I think the most, also in the pharmaceutical field, the most important application area is mode of action screening. That means a very efficient way to get comprehensive pharmacodynamic information on a lot of targets, which are, and pharmaceutical companies have usually much more targets than they can develop, and it's very difficult to select the most promising out of those. And from a biomarker point of view, is also very interested to characterize the adaptive changes which are occurring, because they also need to be have therapeutic need in complex disease conditions, and which I have not presented, but it's quite obvious that also novel markers for prediabetes or early asthma disposition or something like that are unmet needs which can be addressed.